Hello, everyone, and welcome to our Webinar Miniseries, A Beginner's Guide to Single-Cell Sequencing Part 3, Single-Cell Sequencing Data Analysis. Today, we have Dr. Khalid Abu El-Ardat here as our speaker, and he has been working in the field of biology for 17 years and analyzing next-generation data sequencing for 9 years. He earned his master's degree in biology from American University of Beirut in 2006 and a PhD in applied biological sciences from Ghent University in 2011. He currently heads the tech support department at Singular Biotechnologies. Additionally, he works as a bioinformatician analyzing single-cell sequencing, single-cell RNA sequencing data from various organs and species. Hi, Khalil. Welcome to our Webinar Miniseries. And yeah, stage is yours.
Yeah, thank you, Yasmin. And yeah, welcome everyone to part 3, as was said, of this mini webinar series, which is entitled Single-Cell Sequencing Data Analysis, a Beginner's Guide. And this is important to point out the word beginner. So it's really going to go over the basics. So I'm sorry if you have any experience with NGS, this is going to be mostly boring for you, but I think it's interesting to know. And I think even some people who have been dealing with data analysis do not know maybe some of the basics or all of the basics. So it's nice just to go over them. And the second thing I want to point out is that we are going to go through the data analysis, but only up to generating the count matrix because due to time restrictions. So going beyond that is going to take us also a lot of time. It is interesting. So if you are dealing with single cell sequencing, you want to see these beautiful graphs or what's supposed to be done with the count matrix, I suggest you follow some of the training. Maybe we'll give a training also in the future, but for now there are several tutorials also from people who wrote Surat that go step by step what to do, how to load these count matrices and how to do the quality control and analysis. But for now, we are only going to stick to the basics. So going from the first step, getting the raw data out of the sequencer and up to generating your count matrix with which you can work. Okay, so I stole this slide from my colleague, and you saw this already if you had followed parts 1 and 2, especially 2. If you don't remember this, then you are not paying attention, part 2. This is a big difference between bulk RNA sequencing, if you have done it before, and single cell RNA sequencing. With bulk, it was, I think, my colleague even pointed that out. It's like taking all of these fruits that you see in the picture and making a smoothie out of it. You just take your piece of tissue, you extract RNA, you don't care where each transcript comes from as long as it comes from that tissue, and you do the sequencing. In single cell RNA, you're going one step further. You are trying to take back each transcript, trace it back to its original cell. This means that if you're in the wet lab, you have to do something like this. So our technology, we use something called scope chip to sequester each cell in a well, as you can see here. And then you supply what's called a barcode bead. And the aim here is to isolate the RNA that's going to be extracted from each cell after lysis, so that it will bind, the poly A tail will bind to the poly T on the bead, but also you have these two extra pieces of information. You have something called the UMI in blue, and you have the cell barcode in orange. And these are very important. We're going to talk about them now. But just keep this in your head, this illustration. You see a poly T, you have a UMI and a cell barcode. So after that, you're going to generate your library, you're going to send it for sequencing, and after that, you're going to get a FASTQ file. So what is a FASTQ file, and how does it relate to what we saw over there in the illustration? Now, FASTQ file is basically a repetition of 4 different lines. Yes, here some of you might say there are more than 4 different lines, but it's just because of wrapping. So the second line actually wraps into the third because it's just too long. So here we're sequencing 150 base pairs. And so you have 150 letters in the second line, and those just spill over into the third line. And the fourth line actually starts with these Fs, but also wraps into the second line. So what do they all mean? Now, the first line is the metadata line. It just has information that are relevant to the sequencer, but also to you and to the downstream tools. You have the machine ID. This is good for traceability so that you can see on which machine this was sequenced. You have the run ID. This is just an increasing number, so it increases in increments of 1. Every time you start a run on your machine, on the Illumina machine, it's going to increase by 1. So this was run 284 on this specific machine. You have the flow cell ID. This can also be traced back to which kit you used. Which lane was used. So in this case, it's lane 4. You have which tile number is on this flow cell, and then you have the coordinates of your read. So you have an X coordinate and a Y coordinate where it exists on this flow cell. This is also important because in this case, what I didn't mention here is that we do paired end sequencing. So once we have the library we send for sequencing, we are sequencing paired end, which means each let's say read is sequenced from both sides. And for that, you generate 2 files. Usually they're designated as R1 and R2. This is R1, this is the first one. And in R2, you're going to have your mate, and this you can track down by the coordinates. So you have pair member, this is pair member 1, the other one is going to be pair member 2, and you have the index, and the index is just a multiplexing index. This is when you want to pool several libraries on the same flow cell. You use this index, a multiplexing index. Then we come to the meat of the story, and this is the second line. This is your sequence, and you can see. So before we had the poly A or the poly T on the bead, and you can see it over here. It's marked in blue. This is your poly T. In red is your UMI. And then the barcodes are actually 8 base pairs, 8 base pairs, 8 base pairs. So each green mark is an 8 base pair, and they all generate the barcode, and they are separated by a linker, which is in yellow. The other important part of the file is the quality. So on the fourth line, you can see there are these many Fs, and then there are weird colons and commas. Those are the quality scores. So each base, so if we have 150 letters up there, so 150 bases, we have 150 quality scores. Each base has its own quality score. How are we to understand these quality scores? What is an F? Is it good? Is it bad? So basically the quality scores are FRED33 quality scores. They are minus log 10 of the probability of this base being incorrect. Therefore, the higher the number, the better the quality is, which means there's a lower chance of it being a false base. The encoding is just done using a base 33 ASCII code. What that means is that you take your quality score, let's say you scored a 0, you add 33 to that, and then you convert that to an ASCII. So ASCII is just every letter, every number, whatever you type on your computer has an ASCII conversion. So you get kind of a code which is translated into a letter or a number. So this is a table that shows you the ASCII code and what it corresponds to. If you see, for example, the quality 0, the ASCII code is 33, and therefore it gets translated into an exclamation mark. So if we are going to track our F, we see it towards the end of the table, the last column, F has an ASCII code of 70, and that translates to a quality score of 37. So that's quite high. Why are you supposed to beware of the base 64 ASCII code? This was an old method that Illumina used one time. And they used a different conversion. Basically, you just now add 64 instead of 33. Therefore, you can see you can run into a lot of trouble if you don't know if the FASTQ file was using the FRED33 scores or the 64s, because this is what you would get. So F has an ASCII code of 70, but in this case, you have to subtract 64. You are left with 6, and that's a very bad quality score. So if you are to analyze your FASTQ file, which was generated with a 33 base code with the 64 base code, you are going to get rid of most of your bases because they are going to be filtered out as low quality bases. Okay, so how can you check the quality of your FASTQ? Well, one of the tools that's most widely used is FastQC. It's a Java tool from the Babraham Institute. Currently, it's at version 0.11.9, and it's been stuck there for quite a while now. And what it does, it generates quality metrics for your FASTQ files and then displays them as HTML files. So if we take a look at how what it looks like, you can see that per base of your FASTQ file, so let's say you have 150 bases per read, it's going to analyze your quality score metrics and then generate such a graph. So you see, for example, the first one, and these are what it calls examples of good FASTQ files. I have to say, I've never seen this before. So you will never get such clean results, basically, because they're using, I guess those were generated at the very beginning of Illumina sequencing. You can see that the reads are very short. They're at most 40 base pairs long. This used to be standard for Illumina sequencers, is that you would not sequence very long, but now it's becoming more common that you would have 150 bases or 100 base reads, and you can even go up to 300 or 600 bases in your sequencing. Therefore, the quality will suffer. So you will start seeing in the rightmost graph, you see per base, you have your quality scores as a box graph. And you see already at base 40, you have a dip. And this you will start observing the more you sequence. The longer you sequence, the quality is going to dip. This is known for Illumina sequencers, is that the quality, the longer you sequence your reads, the worse the quality is going to be along the length of the read. So I've never seen this, and because I guess it was written at that time when the reads were supposed to be short. Nowadays, if you run FastQC, you're going to see a few warnings for some of the metrics, but that doesn't mean your sequencing is wrong. It's just standard for FastQC. Another tool I would like just to highlight is a nice one. It's written by Python by Phil Ewells. It's called MultiQC. And it actually aggregates all the quality control data that's been generated by all your tools. So not only FASTQ files, it can also generate, it can also aggregate things related to your mapper, to Picard tools and whatnot, and then will generate an HTML file with very nice interactive graphs. So if you have written a pipeline with so many quality control metrics, you don't want to go through them one by one, just run MultiQC on them and it will just aggregate all the data together. That's quite handy. So now we talked about the FASTQ file. This is what you get from your sequencer. This is also common to bulk RNA and single cell RNA. As I said, single cell RNA has two features that are important. Barcodes is one of them, and UMIs is the second. So what is a barcode? A barcode is basically just a nucleotide tag that you can add to your reads. And in this case, a barcode serves one purpose, in that you are able to trace your transcripts back to the cell of origin. So you can have random sequences, you have semi-random sequences, you can have shuffled sequence segments. In this case, we are using semi-random sequences. And the barcode in this case, so we said the barcode is able to trace your reads or transcripts back to the cell of origin. So that means one barcode is actually one cell, but is that always true? And this is something we are going to talk about towards the end of the webinar. A second problem is that you always have to use your barcodes therefore a 3' mRNA bias ensues. This is, of course, common to bulk RNA sequencing in general. Wherever you use poly A tail enrichment, you're going to have this 3' mRNA bias. But in single-cell sequencing, the bias is even more pronounced. That's because you always need to take your reads back to the barcode of origin. If you lose this connection between your barcode and the read, then it's kind of useless because you don't know now where the transcript comes from. And the third thing is that you have to deal with the errors. So, of course, the experiments are not 100% perfect. Whenever you are dealing with DNA or RNA nucleotides, you always have to deal with some errors that are going to happen along the way, either from the environment where the base changes are going to occur just due to oxidation, PCR errors when you are amplifying your libraries, or even sequencing errors. So sequencers are, of course, not error-free. They're also going to introduce their own errors. And those are things you have to take into account when you are dealing with your barcodes. So if we go to the UMIs in this case, so the barcodes, let's say roughly equal a cell. UMIs equal a transcript or a unique transcript. So this is what UMI stands for. It's a unique molecular identifier. The problem is when you're sequencing, you have to sequence to a certain depth. Depth means how often are you covering a certain base. So let's say the human genome is around 3 billion bases long. So if your sequencer generates 3 gigabases, you are going to have covered your genome once only. But that is not enough. Why? Because we are a diploid species. Covering it once is going to anyway, you're not taking into account one of both alleles. You have to deal with sequencing errors. You have to deal with so many things. Therefore, you do not cover the genome if you're doing whole genome sequencing once or twice even. You cover it X number of times, let's say 30 times or 50 times. The problem is once you are doing this, first of all, you are amplifying your library. This is one of the things you have to do when you're generating your libraries. You have to do PCR. Therefore, you have PCR bias. As you know, not every part of the genome is efficiently PCR compared to the next. There is a bias sometimes depending on the GC composition of your genome and whatnot. So you have also sequencing errors and you have also PCR errors. Therefore, you do not, when you want to count the expression of a gene, for example, of interest, you do not count the reads themselves. You actually count the tags, this unique molecular identifiers that you add to the transcript. And this is what you consider. So in this case, UMI is a tag attached to DNA molecules. It's usually 6 to 12 bases long. So what we are using, what I showed there is 12 bases long. So you have 17 million possible, around 17 million possible 12 base tags. And you can add them by ligation or TS-PCR. So let me show you why this is important. So let's look at this figure here. So let's say the green line is one gene and the black line is a different gene. If you don't take the UMIs, which are little colorful circles on the edge here, if you don't take that into account, you're just counting how many reads cover this gene. You're going to say, well, this gene is really highly expressed, and it's even higher expressed than the black gene. But actually, if, for example, your PCR just over-amplified this gene over here, the green one, you have a problem. You are being biased. You are seeing something that actually doesn't exist. And therefore, it's important to count the UMIs themselves. So if you count the UMIs, you only have 2 UMIs in the green one, but the black one has 13. Therefore, the black one is more highly expressed than the green one. So this way, you just eliminate biases that you have introduced just by amplification, for example, and you are just counting the unique transcripts, taking them back to their molecule of origin. So now based on this, if you take the data of the barcode per cell and you take the UMIs, you can add them to the header of your FASTQ file, and now you can generate basic statistics. You can see how many cells did you capture by counting your barcodes, and you can also see per barcode or per cell what was the gene expression by counting your UMIs. However, in this case, you still have not looked in the biological data, yeah, because we only are looking now. We looked at the first file, so this is the R1, but we did not look at the reverse, at the R2 file. Now, the R2 file contains the mRNA data, so it contains the sequences, the RNA sequencing, and in this case, this is where you are going to get your biological insights. So you checked how many cells you have, you check the UMIs, but now you want to see, okay, what type of genes are highly expressed in which cells. Now for this, you would need, this is one of the most computationally expensive parts of the workflow. This is why mostly you cannot run, for example, Cell Ranger or CellScope on your computer. It's because of the aligner. So the aligner is going to take all your reads and try to find where they exist on a reference. And if we are dealing with human data, then you are going to map to the human reference. So let's build something basic. You can, if you have a FASTQ file with CRISPR, let's say you have guide sequences, you did the CRISPR screen, you have some guides, and you have a reference file containing the guide sequences. So what you can do, you can run something like this. You can have a command in bash that's going to extract your guides from the FASTQ files and attempt to match them to your reference file. And then it will also generate counts. So per guide, how many counts are left per condition, and then we'll generate like a little distribution curve. This is a nice idea, but it has a lot of caveats, a lot of problems. One of the first problems is that you are going to output only exact matches. And this is not good at all, because one of the things I mentioned, first of all, we are diploid creatures, so we will have two alleles. Therefore, you're going to throw out a lot of matches just because they map probably to the other allele. The second is that we have SNPs between humans, so we will not match the reference genome 100% naturally. We have mutations and we have the sequencing error. So from the get-go, you cannot do 100% match. In this case, also our reference was very small, and therefore doing something like this is not going to work for a bigger reference, bigger genome. So we have mappers or aligners, as they are called, and there are two types, basically two broad categories. You have de novo, which is reference-free. You do not rely on a reference. You try to assemble your reads without a reference. But luckily, we are dealing with humans or mice or rats, and those are very well characterized, so we can use a reference. Another problem with that method is that we are using local alignment. We are using global alignment, but that will not work. So that is good when you have, for example, very small ones. So like BLAST, for example, you take one sequence and you are trying to find a match. But if you are dealing with a huge amount of data and a huge reference, you cannot just try to match the whole length against the reference. So what most mappers do is that, first of all, they, instead of using the FASTA file, so the FASTA reference file, they use an index. And second of all, they use something called seed and extend, which means they generate a small portion of the read and try to match that. They find all possible matches and then focus on that and use an extend method to find the best match. Yeah, so this is basically how it works. If you have a read that looks like this, you find a seed for it. So ACG matches ACG, and then you start extending and see how many matches or mismatches you get, and then you start scoring them. I will not belabor that point, just to because we have limited time. But I will just say that when you are working with RNA, sequencing, you have now a one additional layer of complexity in that you are working with cDNA, which lacks introns. So because of splicing, if you try to match to a genome, you might have the condition where actually one part of your read matches to one exon and the other part of the read is matching to the next exon, which are usually in the reference genome separated by a large intron, which does not exist here. So one of the mappers that's most widely used for RNA sequencing and which we use also in our tool is called STAR. STAR stands for Splice Transcripts Alignment to a Reference. And it is quite fast. So this is one of the main advantages of using it. It uses a modification of the seed and extent that I explained before. So it maps one seed to a probable region, maps the other part of the read to another region, and then tries to stitch them together and see if this is a good fit. And in the end, once you are done with the mapper, what it generates is the BAM file. So this is an example of a BAM or a SAM file. SAM is readable, so you can just open it in your terminal, for example, and you can read it, whereas BAM is not human readable. And the BAM file, I'm not also going to go over it a lot, but you have some information from the FASTQ file. This is the first part. Then you have the chromosome, you have the genomic region, you have what's something called a CIGAR, a string, which tells you how many matches, mismatches, indels there are. And then you have the mapping sequence. So let's start with the tool that we are using. So it's basically called Celescope. It's found on GitHub. So if you want to use it, it's freely available. You just have to go to the URL down there. Or if you're using Git, you can just clone the repository and play around with it if you have some single-ROM data. So basically, Celescope, the steps it goes through, and this is common to if you are doing single-cell RNA sequencing. So I talked about the barcodes and the UMIs, and the first step is actually to extract these barcode and UMI data and put them in the header of your FASTQ file. So what you do, as I said, there are caveats to using barcodes. First of all, you need you have sequencing errors, you have errors that can happen during library prep, and therefore you have to do some filtering. And in this case, you filter against a whitelist. So we have a whitelist of all possible barcode sequencing, but we also allow for mismatches, and this is this Hamming distance. So any mismatch, one mismatch would be a Hamming distance of 1. And anything that has more than that is going to be filtered out. We'll filter all reads which have no linkers in them. This is the orange part that I showed in the FASTQ file. And all barcodes and UMIs that have a low quality are also going to be filtered out. So here you did the first level of extraction. You extracted your barcode and UMI data. The second would be doing trimming. So you would remove the poly A tail if it's longer than a certain length. So if it's more than 18 consecutive A's, you will also cut it out, and you will cut out the Illumina adapter. So why is this appearing? So usually the adapter shouldn't be there because you usually do adapter trimming. Problem is when you have shorter inserts, sometimes you might sequence into the Illumina adapters. And therefore, if you find these sequences in your reads, you can also cut them out. This will help with the mappability, which is the next step. So you'll have to do mapping. And as I said, the mapper that's used here is STAR. So you would map to the reference genome. If you're using human, then you can use the latest draft of the human genome. If you're using mouse, you can use that. So whatever species you're using, you just have to supply the genome to it. And then afterwards, once you have your BAM file, you can generate a table with the barcode, UMI counts, and the features. The features are extracted from a GDF file, which contains such data as exons or introns, the gene name. One important step, and this is also something that mentioned, is one barcode equals one cell, not always. So, and this is one important step that Cellscope does, is that it tries to find a threshold. Where does it call a cell or a barcode a cell, and when is this just background? And the last step is, of course, generating the count matrix with which you are going to work later. So this shows you the cell versus background. So if you, in the x-axis, you have all your barcodes, you rank them. So barcode number 1, which is cell number 1, has a UMI count, which is a transcript count of, let's say, 50 or 60,000 UMIs. And then you go down. So the second barcode, second cell has less, and so on and so forth, until you reach like an infection point. At this point, where the blue turns to gray, you say, okay, well, actually, this is my threshold. I'm going to consider everything above this point a cell, and everything below it background. And therefore, in this case, I think we had like 8,600 cells, and the rest is noise. And you can see that it's noise because if you look at the x-axis, you see we go up to 1 million. And actually, it is highly unlikely that we captured 1 million cells. So actually, this is just noise. And this is what your cell count matrix looks like. You have just normal 2D matrix. The rows are your genes. So each row is going to be one different gene, and each column is going to be one different barcode, which is considered a cell. Why is it called sparse matrix? That's because, as you know, we sequence to a certain depth. We try to have each cell having 50,000 reads, meaning that a lot of moderate or even low expressing genes are not going to be caught here. So either the gene is not expressed, or it is expressed in such a low quantity that you do not see it in a certain cell. And therefore, for a lot of genes, or most genes in each cell, you're going to have a count of 0. That's why it's called the sparse matrix. And you have to find some ways of dealing with that downstream. But of course, this is a talk for a different day. So in the end, what you have, of course, is trying to export it. And you have several kinds of formats. So the most common, or relatively common, is the one generated by the 10x format. And those are 3 different files. They're tab-separated files, classified as barcodes, matrix, and genes. And they are usually found in a tart folder. So if you are looking through data, you have a paper and you want to download their data, most likely if they use the 10x, they're going to have their data in this tart folder format. You have also the H5AD, which is a compact format of these sparse matrices. You have a WOT, which is actually inverted, so you have to be careful when you have this format, or HDF5. So the field is still, it's still growing. It's still, let's say, in its childhood. So you do not, there are still many formats out there. And we have increasingly more, let's say, packages, bioinformatic packages that are dealing with converting between these different values. Which is sometimes annoying for some people, of course, because if you get your matrices in H5AD and you want to use Surat, and they tell you, oh, you can easily import 10x format, then you have to sit and find a way to convert your data in a way that can be read into your tools of interest. As I said, there are more and more tools coming out that are dealing with converting, but usually you'll have to go through an intermediate. So for H5AD, you can convert it in R using one package. It's called Surat disk, into an intermediate file format, which you can then read back into R and then go on with the rest of the analyses that you want to do. And yeah, this is where our journey stops. So now you have your count matrix and you can sit and play around with it. As I said, there are many tutorials that explain to you what to do with these count matrices, how to normalize them, what type of QC to do on them, and then you can then go on and just visualize, you can annotate. So yeah, as I said, you can go out and try it for yourselves, or you can wait, and maybe we have a training on that as well in the future. So yeah, thank you for your attention. And yeah, if you like this webinar series, then keep an eye out. We are always having different types of webinars. You can follow us on social media and check what we are posting. And yeah, if you have any questions.
Yeah, thank you for the nice presentation. I think we can start our Q&A section now. If you have, so feel free to ask your questions. I can see some questions here. So let's start. The question is every UMI considered unique?
Yes. So, of course, the caveat of using UMIs is that you have to use enough UMIs that you are sure that each UMI is going to tag one transcript. So that's why they are increasing in length. So if you have 6, you can calculate, and not every UMI is a hit, right? So you cannot do a UMI that's just A. Therefore, you have to first of all do a pre-selection. What possible UMIs do I have when I have only 6 bases, and how many molecules am I starting with? You just have to make sure that your UMIs are much higher than the amount of transcript you are starting with. If you have UMI possibilities that are less than your initial transcripts, then yes, you have a problem. That not every UMI is a unique transcript.
Yes. Next question is, how does Telescope distinguish between cells and background?
Oh yeah, that's a good question. How do we determine the threshold? Basically, what we do is, because you know how many cells you loaded onto the chip and how many cells you are expecting, you can take this number and then you take the 99th percentile of the UMIs based on the barcode, like this barcode. So let's say you have 3,000, you take the 99th percentile UMI count and then take 10% of that. That would be your threshold.
Okay. And yeah, the last question is if, so actually they ask if our service lab also covers secondary analysis.
Yeah, that's also a good point. So basically, if you send your samples to us, they will be analyzed up to the gene count matrix. So what I presented here is all included in the package. Now, if you mean by secondary analysis downstream, that also we do, but it's not part of the standard package. But we also do the secondary analysis. Yes, that's no problem.
Okay. Yeah. So maybe if there is any other question, just feel free to ask. Yes. If not, then I think we can finish this. The part 3, the last part of this webinar mini-series. So thank you for joining us again, and feel free to ask us if later you come up with any question, just feel free to contact us, and we'll be glad to support you. Thank you so much, and have a great day. Bye.
Yeah, thank you. Bye.